Now let's get into the details of the adaptive immune system. First of all, let me tell you about the antibodies. Antibody molecules can act as both soluble and cell-bound receptors. First of all, let's discuss the B lymphocytes. On the B lymphocytes, the antibody molecules synthesized in the cell are exported to the cell membrane, where they recognize small components of proteins, carbohydrates, or other biological macromolecules such as antigens, and are taken into the cell to start the triggering processes. Each B lymphocyte is programmed to make antibodies of one single recognition type out of a possible hundreds of millions. When the B lymphocyte is triggered, large amounts of its antibodies are secreted to act as soluble recognition elements in the blood and the tissue fluids. This is referred to as the antibody response. Antibody in serum is often referred to as immunoglobulins, abbreviated as an ING. Some cells possesses the Fc receptors that allow them to take up the antibody, insert it in their membranes, and thus become able to recognize a wide range of antigens. This can basically greatly improve the phagocytosis, but can also be responsible for a lot of allergies. Now let's take a look at the T cell receptors. T lymphocytes carry the receptors that have a similar basic structure to an antibody or B lymphocytes, but with important differences, such as they are specialized to recognize only small peptides, basically pieces of proteins that are bound to the MHC molecules. Secondly, they are not exported but act only at the T cell surfaces. Lastly, we have the MHC molecules. These come in two types. MHC class 1 molecules that are expressed on all nucleated cells, while class 2 MHC molecules are normally found only on B lymphocytes, macrophages, and dendritic cells. Their role is to present small antigenic peptides to the T cell receptors. The class of MHC and the type of T cell determine the characteristics of the resulting immune responses. Their name comes from their important role in stimulating transplant rejection, which we'll be discussing in the upcoming lectures. Then we have the natural killer cells. Natural killer cells share the features of both lymphocytes and the innate immune cells. They are specialized for killing virus-infected cells and some tumors, and they possess receptors of two opposing kinds. Firstly, the activating receptors that are analogous to PRRs, recognizing the changes associated with stress and virus infections. Secondly, we have the inhibitory receptors that recognize the MHC class 1 molecules. They are preventing the natural killer cell killing normal cells. The result thus depends on the balance between the activation and the inhibition.